HGMD, the comprehensive database on human inherited disease mutations, helps researchers and clinicians identify the disease phenotypes associated with published mutations. The core of the HGMD database is the mutation data, which is easily accessed by multiple search options. These search options, which are covered in other videos within the collection, lead to a mutation report. In this short video, you will learn about the different types of information that are presented. We'll use a disease-causing mutation of the RB1 gene as an example. At the top of every mutation report is information about the HGMD accession that is used to uniquely identify the mutation within the database, the primary disease or phenotype that is reportedly associated with the mutation, HGMD's assigned classification, and the gene that harbors the mutation. To see other mutations that are harbored by the gene, navigate to the Gene Summary report by clicking the hyperlinked gene name. For more information about how mutations are classified within HGMD, please see our video on this topic. Additional information may be provided that is dependent on the type of mutation. In this example, we are looking at a missense mutation, so we see information about the codon change, the amino acid change, and the codon number. For other types of mutations, we may see different information, such as whether a splicing mutation affects the donor or acceptor site, the sequence that was inserted or deleted, or a description of a complex mutation. Notice that every mutation report contains a feedback button in the top right-hand corner of the report. Use this button to contact the HGMD curators when you believe that a mutation has been incorrectly classified, you would like to point out a reference that should be included, or would like to suggest that a specific mutation be added, as well as for general suggestions for new features. Below the top section is a list of the cited references. Every mutation report will have at least one citation, which is the primary literature report that describes the mutation and its association with the reported disease or phenotype. Click the PubMed accession link to navigate to the PubMed abstract of the reference. HGMD curators may include additional notes about the mutation in the comments section. For some mutations, additional citations may be listed. These may be additional references that support the primary disease or phenotype association. Alternatively, they may provide information about additional diseases or phenotypes that have been associated with the mutation, such as in this example. Or they may provide information about additional characterization of the mutation, or even information that refutes the original classification and justifies a change in classification. The new insight that the reference provides will be described in the comments section. Below the literature citations is a section that provides additional information about the mutation. Depending on availability, this section may provide the genomic sequence of the coding strand, the genomic coordinate for the mutation relative to the HG38 reference genome, links to external genome viewers, such as UCSC, the representative RefSeq transcript and protein with the mutation described in HGVS nomenclature, description of the mutation in VCF format, links to protein structure information, the dbSNP identifier and link to the dbSNP database, a repeat of HGMD's classification for the mutation, and information about whether the mutation is present in a CPG region. For those mutations that have been mapped to genomic coordinates, information relative to the HG19 reference genome can be obtained by clicking the Legacy Data toggle. Although it's not shown in this report, if a mutation has been reclassified at any time since being added to the HGMD database, a variant history section will be displayed that provides information about the change in classification. The last section of the mutation report is specific for missense mutations. It provides information about the comparative physical properties of the amino acids encoded by the reference and altered codon, information about the degree of conservation of the reference amino acid across a diverse set of species, as well as algorithmic predictions of the likely effect of the mutation. For questions about the mutation report or any other feature, please write to us at bioinformaticssupport@qiagen.com.